Hi, my name is Danielle Brianna Ingram, and I am a freshman here at North Carolina A&T, and today I'll be going over my protein project for Unit 2A. The hemoglobin protein is a very, very important molecule in not only humans, but animals as well. The hemoglobin protein is responsible for carrying oxygen from the lungs to the rest of the body, or from the gills to the rest of the body. I classified hemoglobin as a transport protein. I decided this was suitable because hemoglobin is responsible for the transportation of oxygen. Let's get into the location of the protein. The hemoglobin protein is normally found in humans and some other vertebrae. Hemoglobin is often found in the red blood cells of the organism. In fact, it makes up close to 97% of the red blood cells' dry content in mammals and 35% of the total content of a red blood cell. Although hemoglobin is present in all humans, the actual mutation is often found in African American people, thus meaning African American people are more likely to be diagnosed with sickle cell anemia. This is actually due to African ancestry, which often consists of the sickle cell trait, which protects people from dying from malaria. The hemoglobin protein becomes less active. Once the mutation occurs, the hemoglobin protein still tries to take in oxygen. However, it doesn't take in as much oxygen as it would in a person without sickle cell anemia. As time goes on, each red blood cell starts to carry less and less oxygen. Due to the cell lacking oxygen, it becomes less flexible, forming a sickle cell shape, and begins to block the capillaries in the body. This also decreases oxygen flow. Now I'm going to explain the differences between the original hemoglobin molecule versus the mutated hemoglobin molecule. The main functional group on the original amino acid side chain is the heme group. The original amino acid side chain is hydrophilic due to the presence of the glutamic acid, which is polar and negatively charged. The size stays the same compared to other amino acid chains. The original amino acid can participate in two different bonds. Due to the hydrophilic property of glutamic acid, it is apparent that glutamic acid can form a hydrogen bond. Glutamic acid can also form ionic bonds with any molecules that are positively charged. The properties of the new AA side chain are pretty different from the original. The mutated AA side chain contains valine instead of the glutamic acid. The functional group now present is methyl instead of the heme group. The mutated side chain would also become hydrophobic and nonpolar. It is also negatively charged, which is definitely different from the original, which was polar. The mutated chain would make the hemoglobin molecules stick together, forming long chains. The bonds in the mutated amino side chain would form nonpolar covalent bonds, which are weaker than the bonds in the original. The mutated amino acid side chain can also no longer create the hydrogen bonds. This is also because it's hydrophobic. The mutation affects the protein function, the function of the cell, and also the function of the organism. The mutation affects the protein because the protein can no longer carry as much oxygen as it used to. It also switched out that amino acid. Because the amino acid can no longer carry as much oxygen, the cell cannot either. Because of this, the organism has a lack of oxygen and become tired much quicker than the average person without sickle cell anemia. For the last section of the video, I'll talk about the model that I created for the hemoglobin protein. This is the original, and this is the mutated. So the mutation itself is actually only a point mutation. This means one of the amino acids is swapped out for a different amino acid, thus changing the structure of the protein. The actual cell also changes and becomes sickle due to the lack of oxygen, like mentioned earlier. So let's compare my models. This would be my original model with the amino acids. This is actually the primary structure of the hemoglobin molecule. So I'll point out the 2 amino acids that I believe are vital for this project. Would actually be, this is number 6 of the amino acid chain. 1, 2, 3, 4, 5, 6. So this is actually the glutamic acid. However, once the mutation occurs, you can see that the red has now switched to blue. This would be my mutated model. Because the red switched to blue, it means basically that it's valine now. I hope this sums up everything for the hemoglobin protein. Thank you for watching.